welcome back to my channel. In my previous video I covered characteristics of chordates. I've also done a video on characteristics of vertebrates. I'll link them both down below, and in this video I'll be going over invertebrate chordates. Now if you like this video, please leave a like and comment down below any suggestions of anything you wish for me to cover in the future. Now, jumping straight into this, there are three sub-phylums of Chordata. They are Urochordata, Cephalochordata and Vertebrata. Now, seeing vertebrates are animals within the Vertebrata sub-phylum, in this video we are going to go over the other two sub-phylums. Now, the first sub-phylum is Cephalochordata. These have several characteristics such as a closed circulatory system, meaning they do not possess a heart, but the circulatory system is organized around two major vessels. They also don't have any type of vertebral column, like invertebrates or any structure anterior to the notochord that might be recognized as a distinct cranium or head. Other characteristics include large branchial baskets used for filter feeding, persistent notochords, and have a single copy of many vertebrate genes. Cephalochordates, like adult Urochordates, just sit and filter through most of their lives. They don't attach to a surface, but instead bury inside mud and sand, with only its mouth and the tip of its snout protruding out of the ground. Now, Cephalochordates have long been thought to be the sister group of living vertebrates. In craniates, the living Cephalochordates show many highly derived characters, such as an elaborate anterior feeding apparatus. These have, however, been recently researched, which suggests that Cephalochordates have a much simpler genome than vertebrates, with only a single copy of most family genes. Now, most vertebrates, however, have 4 or more copies of most of their genes, which is probably a result of polyploidation in a basal lineage. Now, moving on to the second invertebrate chordates. Now, these are the Urochordates, known more commonly as tunicates. Now, these are more familiarly known as sea squirts, and have an oral siphon which carries a feeding current of water. Now, tunicates are also quite genetically similar to vertebrates, such as the gene Brachyury. I probably pronounced that wrong. These days it is considered that Urochordates are one of the closest relatives to the craniata clades. An example of Urochordates is from Paleocrasus, and Urochordates possess this oral siphon, which they use as feeding currents, which they pump through a ciliary action in small slits in the side of the pharyngeal chamber. These secrete mucus in the pharynx, covering these slits, trapping the food particles from the water currents. Now, their entire pharynx is enclosed within a loose tunicate, hence how they got the name tunicates, which trap the filtered water inside of their atrial cavity, which they discard outwardly through the atrial siphon. Now, some Urochordates develop directly from eggs into small sessile tunicates. Now, sessile meaning that they are immobile and are fixed to one position. Now, many others have free-swimming tadpole-like stages, which undergo metamorphosis to generate these very different body plans to the adults. Now, adults demonstrate very few characteristics of chordates, but are still considered within the chordates due to this tadpole larva's characteristics. Now, Detailed explanations of the structure and development of larvae revealed remarkably similar to that of the vertebrate embryo, possessing a dorsal nervous system and a structure more closely resembling a notochord with muscle blocks as a tail. If organization of ascidian tadpoles and vertebrate bodies are both derived from a common ancestral structure, then it would be expected that homologous organs in the two groups have been built in similar structures. For example, we can expect the characteristic cell types of notochords to be specified by the same genes in both groups, whereas if the notochord had an entirely different evolutionary origin, then there is no reason why notochord cells should be specified by the same genes. There have been experiments to test this. The results show that one gene seems to play a primary role in the specification of vertebrate notochords in a developmental regulatory gene called brachyury, literally meaning short tail. In fish mutant brachyury, no notochord developed, the body doesn't elongate and doesn't have a tail. Closely related genes is expressed in a notochord of ascidians, and when expressed in other cell types, the ascidian tadpole can morph into notochord cells.